webinar. So today I want to introduce TomoAnalysis, how it works, and what is the background or the running principles for the analysis. And then I'll briefly show how the workflow goes in TomoAnalysis. So let's see how the TomoAnalysis makes the analysis. So this is the typical data that acquired by the color tomography instruments from Tomocube. So basically it's three-dimensional and it's label-free, and it should be correlated with fluorescence signals to have better understanding about the sample. And at the same time, we can take time-lapse data. So we can say the data is very multimodal and very complex. So we need to make a dedicated or an analysis software that is comfortable to deal with this kind of data. And at the same time, we know that we have released a precedent of the current HTX1 system, which is HT1H or 2H. So we need to deal with these kind of data as well. So we have made a new software based on these kind of requirements. So let's see what kind of analysis softwares are at the market. The first thing for microscope manufacturer is the first software to have is dedicated software for their own modalities or imaging techniques. So it should be optimized for specific imaging modalities and workflows. So in terms of when we say about our holometric data, we need to have some requirements to deal with those kind of data. And there are a bunch of things like light sheet microscopy and confocal microscopy, which is specialized for typical manufacturers. So for each categories of modalities, they have built a dedicated software to deal with those kind of things more conveniently. On the other hand, there is open source software, which is ImageJ or Napari Cell Profiler and so on. They are very well known for its flexible analysis because there are a variety of kind of images that is to be dealt with. So they made it very flexible to have more flexibility and more accessibility about the data characteristics. And because of the open structure, the contributors contribute many diverse techniques, very novel techniques. So you can access very novel techniques with that kind of open source software. So when it comes about the novelties or a new analysis techniques, it's very convenient to access those kind of things. And on the other side, there's very genuine programming languages. So some people can use Python, Java, C++ to have their own programs. The characteristic of this kind of software is it's very time-efficient analysis, very powerful for a very specific and repetitive task. So some people are dedicated on some very specific targets, then they build their own programs to have more suitable or efficient analysis. But in a normal, typical, flexible analysis, it's very limited to use those kind of things for more general biologists. So we understand these kind of requirements and demands. So we developed TomoAnalysis to reflect these kind of requirements. So it's dedicated for holotomography, which is 3D. And some people want to analyze the data in 2D or in MIP. So we adopted techniques for 2Ds as well. So it can deal with 2D or 3D data in holotomography. And we incorporate modular approach for flexible analysis. So you can combine multiple or different kind of processes, which we call processes for each modules. So we can combine multiple processes to build your own or very specific to reflect a specific requirements or specific targets. And we also incorporate our proprietary codes for efficient analysis, which uses GPU to enhance the efficiency of analysis. So this is the basic idea how we developed TomoAnalysis. And let's see how bioimage analysis looks like. So this is the typical analysis procedure for quantitative analysis. So the important thing here is quantification. And for quantification, you need segmentation, which means to define the area or region of interest for your own analysis. And for this segmentation, you require pre-processing and post-processing to have a better refined definition of the region of interest. So for segmentation, the most well-known or the simplest form of segmentation is thresholding in fluorescence. So by selecting a certain value, which is called threshold value, when some of the parts of the image is higher than the threshold value, it is considered there is some targets. And when the value is smaller than threshold, we call it we think there is nothing. So by using the threshold, we can define the segmentation area, like in the middle. And we see small holes here, which is not desirable. Then we can get some kind of post-processing to fill these holes. So by using fill hole process, we can fill up those dark spots here and have these nicer boundaries or regions for the analysis and take the quantification for these regions. And before segmentation, we can also apply some kind of pre-processing like denoising or Gaussian filters and so on to have a nicer image for the segmentation. So this is the simplest form of the analysis procedure: pre-processing, segmentation, post-processing, and then quantification. So these processes are done in TomoAnalysis. So based on these basic steps, we have classified 3 strategies for segmentation. The first one is fluorescence-based tagging. So most biologists are familiar with fluorescence. In fluorescence, they usually use thresholding. So by using thresholding, they can take the area of the specific labels, and by using that, they can easily get the quantification results. On the other hand, when it comes to the holotomography, it's non-specific. So we should take some more criteria to get the uniform object. But the basic idea is very similar with fluorescence. So it uses holotomographic image, not the fluorescence image, but the idea is very similar. So in this case, we captures the unique RI features like the RI value or its gradient or its relative intensities, relative change across the region. So based on that features of the object, we can get the segmentation of the uniform object. The examples are like this: lipid drumlets or mitochondria. So we know that mitochondria have substructures, but in holotomography, the resolution is not enough to resolve those tiny structures. So in holotomography, it's rather imaged as a single, it's kind of strand. So it's quite uniform in terms of holotomography, and we take advantage of that uniformity to get the mitochondria and segmentation. And the other part is object with an internal structure like cells or nucleus. So the structures like cell or nucleus, because they have very complex structures inside the object, it's very difficult to get the segmentation with a few criteria. So it requires complex and context-based segmentation method. So that's the hardest part in segmentation. But there are a few ways to get those objects segmented. So we will briefly see those results as well. So this is how the segmentation happens in TomoAnalysis. On the left side, it's fluorescence tagged object. So in the left side, the nucleoli is labeled with a kind of fluorescence dye, and we can segment those areas based on the fluorescence signal and get some quantification results. And on the middle, in the middle, you can see the lipid droplets segmented by TomoAnalysis. You can see bright dots here, whose result is displayed on below. And on the right side, it's cell segmentation. By capturing the Cs of the cells and expanding the Cs for each cell, we can get these kind of segmentation results and get some volume and other geometric information from these segmentation results. So these three strategies are incorporated in TomoAnalysis. And the good thing is these all segmentation is based on the tiny blocks of which is called processes. And by combining those processes, we can get these kind of segmentation pipelines, analysis pipelines. So it's very flexible. So by combining these processing, segmentation, post-processing, quantification, by combining these processes, we can get strategies for various targets. And the good thing is, by mixing or by combining those strategies, we can have more complex analysis, like multiple targets at the same time. And we also can do some relational analysis. For example, like when we segment or analyze lipid droplets, we can get the segmentation of LDs. But at the same time, we can segment those cells. And by combining the two segmentation results, we can get the LD quantification per cell. So by this way, we can extend the analysis scope for more complex ideas. So this is one of the examples of the combined analysis. So here on the left side, it's tagged with two different kind of fluorescence. One targets for the nucleus and the other for the nucleoli. And by segmenting those two organelles or the targets separately, we can get these masks. So one for the nucleus and the other for the nucleoli. And by mixing the two, we can get this kind of analysis. So here, 1, 2, 3 means the nucleus, and here, 1, 2, 3. And from this, we can see the average Li value of the nucleus is very similar, but the nucleoli value differs depending on the cells. So by this way, we can extend the knowledge about the targets using fluorescence and the holotomography. And the other analysis combination is using fluorescence and HT for like drug quantification, drug intake quantification. So on the left top side, it's fluorescence labeled drugs. We treated the cell with drugs and take the fluorescence image like this. So we can see that the drugs are inside the cell. And at the same time, we can get the segmentation of the cells. And by combining the two, we can get the drug intake per cell, depending using these two combination of these two analysis results. By this, we can get this kind of advanced analysis by combining the two strategies. So this is the example how the TomoAnalysis can be configured to have very diverse type of analysis. So for this kind of diverse analysis, we have incorporated a myriad of processes for pre-processing, for segmentation, for post-processing, and for quantification. So there are more than 100 processes at the moment, and we are adding processes as we need and as as TA evolves. So once you use TA TomoAnalysis and find some features or processes are lacking, then just let us know to have those features or those processes for better analysis. And this is the pipelines that we have found useful for analysis. So one for fluorescence, so we can extend the channels from 1 to 2, and lipid drumlets, and instant segmentation for cells, and those fluorescence intensity measurement for the cells, and lipid droplet analysis for each cell. So by combining the three pipelines on top, we can extend this kind of advanced analysis based on those basic segmentation analysis pipeline. And the important thing here again is the quantification parameters. So because TomoAnalysis deals with 3D information, we give a geometric and physical information based on 3D. So the center point and the center of mass in 3D, and volume, surface area, projection area, sphericity, diameter, ferrite length, and so on. And for physical parameters, there's dryness, concentration, and the mean RI or max, mean, and range. Such kind of statistical information about the distribution of the RI value. So we can extend those geometric and physical values as we evolve the TA. So let us know if you want more parameters to add on your analysis. And after the analysis, it's important to have a visualization. So based on the analysis results, you can extend the analysis in 3D. So based on the segmentation, you can crop or the focus, or you can focus on the targets. Here we acquire the cell segmentation, and based on the results, we can get this kind of visualization in 3D. So that's the benefit of having 3D analysis. So based on the analysis result, we can have this kind of three-dimensional visual rendering as well as quantification results. So let's see how the TomoAnalysis works in reality. So the first thing that we should know about the TomoAnalysis is how the workflow goes. So in reality, we should select the files and select what kind of modalities that we will use. And when the data contains time-lapse data, we need to select the time point. So the second process is select the workset, which we call workset, which defines the channel and the time point for the analysis. And then we can set the region of interest if we want, based for each workset to exclude or include some of the region of interest. And the first 3 steps are for the preparation of the data set. And from 4 to 5 is the actual analysis. So the step 4 is for parameter adjustment for optimal analysis. So when you select pipelines, analysis pipelines, there's some parameters to adjust for better analysis, and that's the step for preparing the analysis pipeline. And then you run in batch, because you can select multiple files and multiple worksets. The analysis software automatically runs the pipelines through the files or the worksets you have chosen. So that's the step for the batch analysis. And after the batch run ends, you can review the results. So this is how the workflow goes for the in TomoAnalysis. So based on that workflow, this is how the step 4 looks like. So when it comes to the segmentation of fluorescence type objects, the data looks like this when you load the data. And by simply selecting these threshold values, like number 2 here, actually for this fluorescence segmentation, it's very straightforward. And the only parameter, usually the only parameter that you need to control is the threshold value. So depending on the experiment, the intensity, fluorescence intensity can vary. So yeah, to reflect the intensity changes, you can adjust the threshold value like this in this second process. Here, I chose 250, for example, and get this kind of segmentation. And if you're satisfied with these settings, then you can set this value for the batch analysis, and you can go with these parameters. So this is example of how the analysis goes. So it's very straightforward. So here I intentionally set a value too high for these images. So you see that some of the nucleus here, actually it's nucleus, the nucleus is stained. So here you can see some of the nucleus is filled and there is no spots in the middle, but some of the areas looks very spotty. So we need to fill. Actually, I applied the fill hole process to fill the holes, but it's very sparse, so it couldn't cover the whole area except these two. So in this case, you need to adjust the parameters to reflect this kind of situation. So you may need, lower the value to include those excluded area here. So let's see how the process looks like for these fluorescence tagged objects. So here there are in total 5 processes. One for pre-processing. So this is Gaussian filter for smoothing. And the second part is thresholding, which is for segmentation. And the third part is filling holes, which is for post-processing. And the 4 and 5, it's for quantification. The fourth process is for labeling, which means after the thresholding, actually the objects are not separated each other, one another. So by assigning labels for each object, we can get the quantification result per each object, And so the fourth process does the job for labeling. And based on the labeled mask, number 5, analysis on target, which quantifies the values for that target. So in this 5th process, the result in RI or volume and other parts of the measurement data comes. So this is how the process for the fluorescence segmentation works. And when it comes to the uniform objects, it's very similar to fluorescence-based segmentation, but it's not done by a single threshold like fluorescence. So we combine 2 kind of segmentation criteria to have better segmentation results over these lipid droplets. So here we incorporate 2 different criteria. One is absolute RI value, which is here like 1.35. So we lowered a value a little lower than usual value to incorporate unusual LDs as well. And besides these relative RI value, we incorporated a relative RI value. So in some cases, cells, when cells condense, cells contract, the relative the refractive RI value gets higher. So we wanted to exclude those unnecessary parts. So to avoid those kind of things, we applied this kind of relative RI value segment criteria. So in this case, we applied 180, which is actually, it should be divided to 10,000 to have RI value. So it's correspond to 0.018 in terms of RI. So those parts whose relative RI value is higher than 0.018 can be segmented with this kind of criteria. So by mixing the two, we can get these kind of ideas. So let's see how it looks like. So when those areas whose RI value is higher than 1.35, it's like this. So you can see these parts are segmented or distinct, segmented as LDIS because it has high RI value. But we know that this is not the LDIS, but some kind of contracted cell parts. So on top of this, we can apply this delta RI value, which means the RI value relative to its neighborhood, and whose value should be larger than 0.018. And in this case, we can get this kind of results, which is more like LDs. And by having the intersection of these two results, we can get nicer LD results like you showed before. So this is how the analysis process goes. Oh, yes. So here you see that the on the left side, it's the pipelines for the analysis of LDs. And on the right side, you can see the outputs in the middle. So the outputs produced by each process is listed here like this. So in total, including the source or the original data files, there are in total 7 data outputs for that is used for the analysis. And if you want, you can visit each layer or each output to see how the analysis went on during the process. And you can refine the results by visiting those outputs and controlling or adjust the parameters for each relevant process. And also you saw that the results can be displayed in 3D, and also the targets like the LDIS can be separately displayed in 3D to have better idea about the analysis results. So this is how the analysis works in TomoAnalysis. And this is segmentation of object with internal structures, which is the segmentation result of cells. So I briefly introduced how it works here. So here you see the cells are segmented in different colors. And the way this is done is by expanding the seed, taking the seed. It's actually not the real seed of the cell, but it's mathematically calculated the seeds of each cells. And by extending the seeds to make it grow, so by expanding the seed, we can get these segmentation results. And after that, we can label those individual cells based on these seeds. So we can get this kind of good, great segmentation results from these pipelines. So this is the same. This is the procedure or the segmentation pipeline is made of those basic processes that we have implemented. So depending on the processes, you can do many kinds of analysis as you wish. And this is the screenshot of the object of these cells, and this is the same. And when you get the analysis results, you can do the same with the in 3D as well. So based on that segmentation results, you can get this kind of rendering in 3D and get more idea. And you can actually, separate those cells one by one based on the segmentation results. That's the benefit of the segmentation. So by mixing those techniques, you can have very diverse analysis. And you can do many more segmentation or analysis based on these strategies. So you can combine many basic strategies to have diverse ideas. So for example, you can get the LD or mitochondria analysis, and you can do the analysis based on cell as well, and you can combine the two to have better understanding. So in this case, you can use the whole tomograph data to get these kind of ideas. And at the same time, you can tagging the targets with fluorescence, and at the same time, you can get the instance of cells with HT. And the benefit of these strategy, fluorescence and fluorescence tagging and using the cell segmentation, is that you can save your fluorescence dyes. So when you use fluorescence, you should tag many things to have advanced analysis. So on top of the target cells, you target the fluorescent target that you are interested in. Other than that, you need to use diverse, many basic fluorescence to stain the cells or nucleus, because you need separation between the targets based on the cells. But when you use color tomography, you don't need that kind of extra stainings. You can use holotomography to get the segmentation of cells or nucleus from holotomography and combine the result with the fluorescence data, so you can get more, you can get clearer fluorescence signal because you save the fluorescence. So that's the benefit of these combined strategies. And you can do the multiple fluorescence experiments and do the experiments or analysis based on the multiple fluorescence, as I shown before. So this is the segmentation strategies that can be done in TomoAnalysis. And you can ask some of the questions about TomoAnalysis. So I added here briefly. The most frequent questions are like these. So does TomoAnalysis handle data from 2H or 1H, which is the predecessor of the HTX1, which is the current instrument that we promote? And the answer is yes. The process in TA can be applied to any type of data, including TCF. So it does not distinguish between the data from 2H and the data from X1. So you can use the data from any instrument that Tomocube has made. And one of the examples that I shown before, the fluorescent data, it's acquired from 2H. So that's the example of the TA can handle the data from the former 2H instruments. And the second question is, am I able to open the analysis results in other programs like ImageJ? So yes, the answer is yes. The analysis result in TomoAnalysis can be exported in TIFF and RAW formats when it comes to if the data is in image format like the holotomograph data and the label data. And the measurement data, which is in tabular format, it can be exported in CSV. So you can open the data in Excel or other spreadsheet for more statistical or more analysis. And third question is, does TomoAnalysis support machine learning or deep learning algorithm? So the answer for the time being is not now, but the AI functions will be added soon. So we are developing, incorporating those algorithms that is embedded as a process in TomoAnalysis. And the good thing is when we incorporate those functions, AI functions, we also thought about incorporating the functions to run third-party programs as a process. So it can provide functions to use other programs in the headless mode. So it can run ImageJ or SAP-Profiler or other Python codes as a process in TomoAnalysis. So when you see the function of AI in TomoAnalysis, you can also see those third-party programs learn as a process in TomoAnalysis to extend the functions for more advanced and more diverse analysis. So this is the three questions that I have made or prepared for frequent questions. And then I'll go into the real analysis based on the software. So can I have, I think I can get some questions from you if there is. So Liu Xuan asked if, I wonder, is there a specific RI value that you would suggest to segment mitochondria? So at the moment, the RI value, the simple RI value does not guarantee the segmentation of mitochondria. Although mitochondria have some specific RI value, the RI value is not specific to mitochondria. So when you use those simple RI value, then it'll also segment those other parts like cell membrane or the nucleoli as well. So we are searching for more better segmentation analysis, and at the moment we have two strategies for that. So mitochondria, the mitochondria, when it is segmented, it's in a fiber. It's like fiber. So the shape of mitochondria can be used for the segmentation. And so by combining the features, the fiber-like feature, we can segment those mitochondria. And yeah, by taking those features as well, like LD, like LD, where the simple RI value does not guarantee the segmentation LD, and we added the relative RI values for the segmentation. For like that, for mitochondria, we can use the RI value for basic segmentation, but for better segmentation, we can apply the fiber-like features for the mitochondria. And actually, we are developing the feature, so we are not ready to release the mitochondrial segmentation pipelines at the moment. And at the same time, we are also thinking of machine learning process for mitochondrial segmentation and other supervised learning algorithms. So we are successful in getting those mitochondrial segmentation using Elastic, and we are incorporating the features. As I said, we are incorporating machine learning features in TomoAnalysis, and we believe that you can also use machine learning features for segmenting mitochondria. In that case, you can, at the moment, you can use Elastic to run as a third-party program, but at the same time, we are aiming to give the function in TomoAnalysis, including the training procedures. So wait for the release of those features for the mitochondria analysis. And also, Mariana Kunze asked that if the fluorescence signal is only at the parameter of the organelle to segment, e.g. at the membrane, can the fluorescence tagged object workflow handle fluorescence of that sort, or does the fluorescence have to be present throughout the interior of the organelle? I'm asking because of the segmentation of yeast vacuoles. So there are two ways to get those kind of segmentation. So actually, it's not that difficult. So if we ask us, we can support. At the moment, we are very open to support your requirement because we are at the phase of developing pipelines. And I think it's very straightforward. So if the membrane is a fluorescence tagged, we can fill those regions. One way is to fill the regions based on the perimeter segmentation, like fill hole. And the other way is using a bit advanced process, which is called watershed. So based on the shape of the segmentation result, we can calculate the inside of the perimeter, and based on that, we can segment, or we can make a better segmentation mask for that vacuole. So I think we can try multiple ways to get those segmentation. And Hussein Kadem asked that my TA software has AI module, and by upgrading it, I will. It's just first.
Sorry, but could you, because it's Songshik, they raised their hand, so.
Okay.
So Songshik, can I? So we cannot hear you. So could you turn on the microphone?
Oh yeah. Okay. I don't think I can make the something can talk. How can I make him speak?
Yeah, there's. Ahram, please just move on to the Hussein's another question. So this is about the lipid drum analysis.
Okay. So there's Suchin Yadev asking, how should I approach the segmentation of nucleoli and quantify changes in nuclear reflective index when the threshold of certain drug-treated cell undergoes alteration? So the most difficult segmentation targets at the moment is nucleus and nucleoli, because the characteristics of nucleus and nuclei is very very special, because in living cells the RI value of nucleus is lower than the cytoplasm, so it's like void in the middle of the cell in terms of holotomar data. So we've tried many ways to segment nucleus and nuclei, but yet we have not been successful. So that's the painful part for us. But when we have those machine learning features, then we think we would be able to segment those nucleus and nuclei. Actually, nuclei is, I think it's easier than nucleus, because when we get the nucleus segmentation, nuclei is just a bright part inside the nucleus. So the most important part here to segment nuclei is to have nucleus segmentation. So if we solve the problem for a nucleus, then the nuclei will be solved naturally. And Hussein Kadam asked, does the software provide the number of LDs per cell? Yes. When we segment LDs, the LD droplets are separate by the droplets. But the thing here is that when the lipid droplet gets smaller and gets aggregated, it's very difficult to separate those aggregated lipid droplets. And it's the same, the situation is very similar in fluorescence. When LDs are stained with fluorescence and the LDs are too small or too close to one another, then it's very difficult to determine each lipid droplets because the signals are mixed, and it's very difficult to tell the boundaries of each droplet. So in our process, we neglect those small droplets because sometimes the lipid droplets can be confused with other small entities in cells. So it depends on your choice, but in the default pipelines that we provide at the moment, filters out those small LDs, which is usually smaller than 0.2 or 3 micrometer. So when you open the pipelines, you can change the settings for your own purpose, but I think it should be enough. And yes, the answer again, if I answer again, you can calculate the number of LDs, and you can make the distribution chart based on the numbers and the size of the labeled LDs. And I wonder, oh yeah, someone already told, so if I address it again, my TA software has AI module. By upgrading it, I will lose this feature. I mean, the new software is only based on RI segmentation. So at the moment, the current version that I showed you is based on RA value only, and it's based on rules. So when you make some rules based on process, it can analyze based on the rules. And you know that the former version, which is 1.3, it has some rule-based analysis as well as AI-based analysis. And we will incorporate those features on the next version or in next coming year. But at the moment, it is not available. So at the moment, you will lose when you upgrade the software to 1.7, but soon you will have the update version which incorporates the AI features as well. But the thing that you should be aware of is that it's about the important factor for AI utilization is the training. So in the new version, we'll incorporate the training workflow as well to have your own your tailored or your specialized version of models for your own analysis. So that will be different. So yeah, this was the question. I think all the questions are answered. So from now on, I'll show the real workflows on the TomoAnalysis. So this is how the TomoAnalysis software looks like. And at first, I show how TomoAnalysis starts. So at the moment, you can run multiple programs at the same time for your own convenience. So this is the list that I have made for the analysis. So here you can see some webinar focused analysis projects. And when you want to make a new project, press this analysis button, and you can reload the previous analysis project from here, or you can make your new projects. So let's make a new one. And you can select the pipelines here. So as I told, we have made 6 pipelines so far. So the first one is based on fluorescence, single channel, double channel, and lipid drumlets, and instant segmentation for cells. And the last part is by combining those strategies. So here, I'll use the fluorescence-based analysis because it's the simplest form, and it's good choice for explaining the workflows. So this is the screens when it started. So you can see the titles of your project here, and here's the pipeline I've chosen. And at the beginning, it shows what kind of data types it uses for the analysis. And because it uses fluorescence for the segmentation and the whole atomic data for the quantification, it requires 2 channels, one for fluorescence and the other for HT. And there you see the processes for the quantification. And this is how the pipeline looks like. And after selecting the pipelines, then there's the nominal workflows. The first thing is to select files for the analysis. So let's see. So fluorescence, we have a few files. So I take 2, or let's see, just 3. So we can we chose 3 files for the analysis. And the next part is to select work sets, which means we need to choose which channels to be used for the analysis.
Sorry for interrupting, but we cannot see your screen now. So I'm looking at some kind of a small window. So so I guess that maybe you should change it. Maybe the share the screen, I guess. Yeah.
Do you see now? Does it work?
Oh, I don't think so.
Okay, let me check.
I guess you should change it, the screen. So yeah, so we can see the your monitor and the small window is
Small window.
Yes, I guess they say your camera sizes, I guess. So
That's strange.
okay, so can we see it now? Ah, I guess we can see it. All right. Yeah.
Do you see?
Yes.
Okay. That's strange.
Yes. So anybody is please just let me know if they're in trouble.
Okay. So let's start from the beginning. Do you see the screen?
Okay, so let's check once again. So is anyone who cannot see the screen now? So now I can see this, and then I think some of them can see the screen, but some of them Yeah. So now, okay. So Liu and then Marina. Yeah, okay, everyone can see this. All right. All right. All right.
Okay.
Yeah. Just let me know if you have any other trouble.
So this is how the TomoAnalysis looks like. So when it started, on the left side, you can see the latest projects that you have dealt with. So you can use those things if you want, but at this time, we'll build a new one. And so in this screen, you can make this, and you can select fluorescence-based pipelines for the simplest form. And you can omit the users and description part. So when it is started, it's like this. So on the left side, the basic information about the pipelines and the structure of the pipeline is shown. So on top, it's the name of the project that we have made. And on the bottom side, there's a name for the pipeline. And below that, it displays the data sources for the analysis. For this particular pipeline requires fluorescence for segmentation and color tomograph data for quantification. So we need to assign those two sources for the analysis. And it's below the processes for the pipelines are listed. So in total, 5 processes will be executed for the analysis. So this is the most simplest form of the pipelines for the analysis, especially for this fluorescence-based analysis. And for the analysis, you need to register the files. So here, you can select multiple files, so like this. So I prepared 4. So this is how we choose the files for the analysis. And the next step is to assign the channels and the time point if there's time-lapse data. Here, there's no time-lapse data, so we just need to assign the channels for the analysis. And this button is to call the workset editor, which assigns the channels. So here, it has time-lapse data. So the first one, it says 6 time points for holotomography and 6 channels for time-lapse fluorescence. So in this, for this data, we can select 6 data sets from 1 to 6. And for this data, we can use select, which selects all the time points like this. And if you don't want all the data to be analyzed, I just chose a single time point like the last one and apply. Then for this specific file, we chose just one slices like this. In this file, it has only one single time-lapse data, so apply. And for this data, it has 27 slices like this. So we can manually type in 20 data, but if we want all the data sets, then just press this button for selection, and it lists all the time points. But usually we just want to have a certain number of intervals for the analysis, for brief analysis. So let's say 10. So by interval of 10, we can select the time points from this data. And as we want to include the last part, so we can manually type in the last point like this. So in this way, you can adjust or you can select manually the time point that you want. And for example, if you have a bad data in the time point 11, then you can select 10th point instead, or 12th point instead. So in this way, you can adjust or you can select the best data that you want if the exact time interval is not available. So in this way, you can choose 4. And this one, oh, it's very, very strange one. And you can use this one is on, there's only one slide, one time frame here. So I just select 1 and press OK. Then all the time point that I've chosen is listed like this. And this is how the work set is chosen for the analysis. And the next part is setting the region of interest if you want. So it's optional. So we can skip at the moment for this particular demonstration. And the next part is setting the parameters or adjust the parameters for optimal analysis for this specific pipeline. So for the adjustment, you need to select a test data for the adjustment. So for that, you can select one of the files for the analysis and make it active to be loaded here. And I think it's very dim. Okay. Oh, it's really dim. And oh, yes. So here you can see the image can be browsed over the Z direction, which means the data is loaded in 3D. And here you can see the fluorescence data is very vague. So I adjust the intensity, like oh, it's too low, like this. So you can see the fluorescence signals clearly by adjusting the contrast. But these areas for controlling the display, so it does not influence the analysis. So it's independent control for the display only. So when you chose the active files here, so in this case, you can just press this button for the test run. So if you press the button, it runs the parameters set by default, and it goes through the analysis. So this is this looks like what I've shown you in the presentation. So here you can see many spots unfilled. So because I chose a high RF segmentation threshold value for the fluorescence segmentation. So the thresholding is in second part. So I decreased the value to like 30. Save. And I try this button. Then it feels like this. And the next part is fill hole. And I apply. Just take some time. Oh, still there's some spots remaining. So I'll change it to like 5, for example. And filling holes. Most of the parts are filled. So you've seen that it takes a bit long time compared to 2D analysis. The reason is that the data is in 3D, so it's quite big. So it takes some time. So we are developing the code more efficiently, but at the moment it's not that fast. So at the moment, please bear with that. So let's see how it looks like when you hover the areas like this. So you see that along the z-axis, the shape of the fluorescence segmentation changes, and it can be combined with the holotomography data to get the volumetric information about the segmentation. So if we are satisfied with the setting, then run the test again. It's like this. So because it's just for training, just skip this kind of imperfection. So I chose this one for the test run, and I can select a different files for the test run as well. But one thing to notice is that it's not in the same experiment set. So the intensity can be different. The settings can be different. So it's not recommended to mix the files in one analysis project, because depending on the experiment condition, the parameters like threshold can be greatly vary. So you should make the experiment condition the same to have a repeatable analysis. But anyway, in this training session, I just run the batch process with these parameters. So this button is for the batch process with the given or the set parameters. And when you press the batch button, it asks for the which data in the process is saved. So you see the CSV file, which is the measurement file, is recorded, and the volumetric data, especially the label of the fluorescence, is saved in this batch process and safely apply. So here, yeah, the threshold value is not adequate. So it's the segmentation is very weird because it's based on the last set of files. And here it's like this. So you see those segmentation areas separated, but because the parameters were not adequate for this particular type of data, the segmentation result is meaningless, especially the object labeling. So after processing these work set, I'll abort the batch analysis in the middle because it takes a long time, and I can show the batch process can be aborted in the middle. Okay, I abort. So it abort in the middle. And although I aborted in the middle, the three work set here is analyzed already, and we can review those results, although we aborted in the middle. To review the data, we can make it active again and recall the results like this. Actually, it's not user-friendly at the moment, but it's for reviewing the results. So by this, you can get the result, which is saved during the analysis process, and you can see those labels again. And here, the good thing is, when you want to distinguish which one is which, you can set this highlight button, and you can highlight the areas depending on the labels. So here, there's labels for each object here, and by selecting the labels here, it can be tracked like this. Actually, when I click this, there's no changes at all because it's in 3D. So in some region, it should be highlighted, but because it's 3D, sometimes you miss those changes. And you can choose the object in the other way. If you click this one, it's highlighted in this area. So you can say which one is which. So in this case, these two objects are separated but considered as one because it's linked in other location like this. So you can see that it meets in other G position. So it's very difficult to think in 3D in human's mind. So when you are when it's not easy to understand, just browse in Z direction to see if it meets in Z direction. And the results here can be rendered in 3D like this. So you can make it displayed in 3D. So you can display the mask only, all the volumetric information, which is HT or fluorescent data. And you can display the volume with the mask. So you can specify those areas to be displayed with these results. Let's see this one. So for the volume, we choose HT, and for the label from fluorescence. If I click this one, those areas with fluorescence will be displayed, but in this case, the fluorescence mask is not that meaningful, so you will see some weird part. Let's see how it looks like. So some of the part was displayed, but you can see the 3D of the HT without the mask, and you can get this kind of images like this. So here I think it's adipocyte, and you can see very big LDs and the small LDs at the same time. So here I think we intended to see the LDs, I guess, but the staining was not perfect at the moment. So it's not a good example for a good experiment, but I've shown you for the purpose of segmenting based on fluorescence. And if you choose another part here, you can see the results. Oh, by the way, you should reload the results here. So here the segmentation is like this, and the intensity was different from the test data. The result is a bit not that good at the as well. Let's see this one. So this one, I guess the nucleus is better segmented. So as I told, if we choose this highlight button, you can highlight which one is which, and it's vice versa. So you can click anything to see the physical parameters for each object. And you can also apply the volumetric mask here with this features. So here, oh, it's very dark, so I'm changing the areas like this. So here, because the mask is only focusing on the nucleus, only the nucleus was shown here. So that's the reason why it's so that's the reason why it does not contain much information about the real data. And because we get the masks, so you can see that the area is labeled, colored with its label, so you can see the highlight. You can see the separation of the each layers. So in this way, you can use the analysis results to have a 3D rendering of your data. So that's the strength of the TomoAnalysis when it comes to the visualization. So this is how the fluorescence-based segmentation works in TomoAnalysis, which is the simplest form. And the next part is the LD. For the LD, I'll recall what I've run previously. So here, I've chosen this one. So here, the process to create the analysis project is the same. So I named the project target as webinar LD and chose the LD analysis pipelines. And here's the processes. The first one, which is generates top-hat image, it's for pre-processing. And the second and third part is for the segmentation. So the second process is for the absolute RI value criteria. And the third one is for the relative RI criteria. And by combining the two segmentation results, we can get the segmentation of LD. And the fourth part is the combining intersection of the two criteria. And the fifth is the post-processing, which I chose for the filtering out those small LDs not to be confused with small debris. And then the sixth and seventh processes for the quantification. So let's see how it goes when I press the test button. So here you can see the layers are stacking up. So this is the source data, and this is the threshold data, and this is the relative RI value data, and this is the intersection data, and this one is the post-processing for size filtration, and the last part is the final LD screens. So if I close up, and here you can see each layer by layer. So let's see how it goes. So at the beginning, only the source file appears. And when you apply the pre-processing for the top hat, it becomes like this. So it gives the relative brightness. So you can see more clearly about the LD. So you can see the bright spot here. And by taking the relative values, relative RI values, we can segment those areas. So from this polyatomic data, we can get this relative absolute RI value thresholding to segment LDs. And from this data, we can get LDs like this. I think the data is not displayed. I think it's a kind of bug. Oh no, yes. It seems, yeah, it's invisible now because these results, mask LD is the intersection of these two layers. So it should be there, but it's not displayed, I think. And this is the intersection of the two results here, which is based on the relative and absolute Ri values. And here, very tiny particles can be LD, and sometimes it can be other particles. So we omit those small particles. So you see small things are excluded. Here, the small means that the size or in diameter is less than 0.5, I guess. So if the particle is smaller than that, then it will be omitted or neglected from the LD segmentation. And from these filtered results, we acquire the LD analysis like this. So this is the final result. So we missed a lot of dark and bright spots here. And if we want to include those regions, then we need to adjust the parameters. So here you can adjust the absolute values and relative values. And I found the value is a bit high. So that means we imposed a high value for a relatively higher value for LD analysis. So if I change to 137, for example, it's much brighter. And for this tuppet, it's like this. So I think we don't need to change the value. And let's make the intersection. So then it's much better than before. So based on these changes, I just changed this one only. So I wanna make it applied through all the pipelines. So I press this test run button again. Then it takes some, yes. Then it looks like this. So you see that some of the part here, I can go down. So you can see the LDs are segmented here, but the LDs are aggregated and regarded as one single object. So I believe someone asked about the LD separation. So as you can see here, it's very difficult to distinguish those particles, even though we see those particles as many particles, and we can manually segment those areas. But the difficult thing is it's in 3D. So when you go up or go down, then the segmentation is not that straightforward. So that's the reason why we don't supply, or the reason why we don't add extra process to segment those LDs, because it's very tricky to segment those particles in 3D. So at the moment, we only erase or exclude those small LDs just in case it is confused with small debris. So like this, we can get the repeat analysis results. And if we have many work sets here, then you can press this batch analysis button to get the ideas. And one other thing I want to show you here is that when you have multiple data, time-lapse data, although I show it in previous samples, I would like to emphasize it again. So if you add another files in the middle, you need to select the work set again. And there are two ways to select work set here. So just press this button will give you the screens for the work set and selection. And this file is what I've added just before, and it says it has 17 time points for this time of data. And automatically it gives the numbers for the timeless from 1 to 17. And by default, it gives interval 1. So if you press select, you can select all the files at the same time. And when you apply, you can see that all the time points are registered here. But if you don't want to analyze all the files, then you can change files and press apply again, then the number changes. Or if you find that these particular file does not contain good data on the 16th, and instead it have good data in 17th time point, you can manually edit the numbers to get your the time point that you want to analyze. So by clicking yes, it is renewed, and then you can press OK. Here I does not specify the work set for the file set 2 because there's already it's set here, but it warns that you missed the settings, and you can ignore because you already have decided or determined the work set here. So press continue. Then you can see the 4 time points are selected here. And by this, you can analyze these 4 file work sets as well. And if you are satisfied with the parameter setting here, you can just click this button to make it analysis in batch. So in this way, okay, again, you can analysis all the data here. And you see that the process goes on here. Yes. And in the middle, if you are curious about which process is processed, there's a small information box here. So if you click this button, you can see the processes that's on the queue. So which file is processed and which file is on queue and what process is running in the middle. So you may abort in the middle to save time. To this one, quit. Okay. Abort. So the batch processes are the pipelines are applied for these 3 files. And if you want to review the results, just double click or make these worksets active again, and you can recall the result here like this. And actually, at this stage, you can also highlight the things here, but it's very tricky to pick one of the file here, so it's not that useful for these LD pipelines. And at this moment, you want to analyze the results and use the data, the mass data for your own analysis to open an ImageJ like that. Then you can get to the the areas. So description, yeah, you can open the Explorer, so you can see the export for the here, and the time of execution for the batch analysis here. When you go into, you can see you can see the name of the data here. So analysis NLD. This one, yeah, you can see the process name here. So each process saves its results. So analysis, as we've processed 3 files, you can see 3 data sets here for the measurement. And the last one is the summary or the concatenated results of the 3 files. So if you want to analyze in batch for these 3 results, you can use these files instead. And also, the labeling masks are available. So because it's in 3D, most of the cases, the label data is in black because if there's nothing, then the value is 0. So in TIFF, actually it's exported in TIFF. So most of the first slice of the image is in black because there's nothing that is segmented. So if you open this one with ImageJ, you can see the difference. Let's see. I'm afraid that the LDs are so tiny, so it's not that easy to be distinguished. But let's try. If it's open properly, so by drag and drop, you can see this one, and let's make it smaller. So you can see this dots here, right? That's here, which is actually labeled. So let's change. Although I changed the label, it's not that. Oh, yeah, you can see it here, from darkest to brightest, because it's labeled. So each label has its own number from 1 to like so many. So it gradually increases the intensity. So if you open these results with other formats, you can see the numbers here. But in this ImageJ, you can see just the brightness changing. So in this way, you can open the results with ImageJ or other analysis software for further analysis. So you can easily combine the results from TomoAnalysis. And at the same time, in the middle, the screen here is captured for your own for your convenience. So let's see how it's not a data actually, it's just a snapshot of the image. So you can use the snapshot data for your own report. So you can use the results for a quick report or lab note. So in this way, you can use the analysis of the TomoAnalysis for your analysis results. So I think this is the basic information about how TA works and how the pipeline works for the analysis. So the fundamental part here is that you can control the TomoAnalysis, the analysis done by pipelines, which consists of multiple processes, and you can combine those processes to make your own. Even though we supply some model pipelines for analysis, you can extend the pipelines if you use the editor, which we provide at the same time. But at the moment, I've not shown you the pipeline editor because there are a bunch of processes to be understood to be utilized. And here you saw that by modifying some parameters, you can acquire a decent segmentation results, especially when it's LD or fluorescence data. So I think that's all I've prepared for today, and I would like to have some questions from you. Is there anyone
Okay, so we're gonna wait the few minutes. Then yeah, so that is we expect this is yeah it took longer than 1.5 hour. All right, so so I like to thank all of the attendees yeah and listen to the webinar and then our demonstration of the TomoAnalysis. So after the webinar, and then we will reach out to each of you once again to discuss about the installation about of the new TomoAnalysis. So if you already have a TomoAnalysis or you have the Tomocube instrument, and then so the license will be free, and then but we need to ask you the information for the registration of the license once again, because the new license of this new software is different from the other forms. So and we would like to provide the installation file and the manual, the brief, the user manuals on it. And then so if the research the design is not is more than the fundamental training today, and then we would like to have additional the one-on-one but the training sessions, yeah, and to optimize it. So I already asked some of the attendees, yeah, about just providing some sample data, because after reviewing the data, and then we can further optimize our standard pipeline, and then we can provide us additional pipeline if it's a simple. So so we're happy to optimize our analysis part workflows suitable for your study design and then your data. So I would appreciate it if you have any question in the meantime, just reach out to me. Yeah. And then if you have any other questions, and then the Ahram will also answer some questions by emails. Okay, so if there is no further questions, and then we are going to the wrapping up wrap up today's webinar. So thank you for today's speaker Ahram, and then I hope that we can talk to you once again in the shortly. Okay, so have a great the afternoon the remaining afternoon, and then goodbye for now.